The protein I researched this semester is the C-reactive protein, also known as CRP. Did you know that cardiovascular disease is the leading cause of death for both men and women in the nation? Fatty deposits can build up in the arteries and cause atherosclerosis. Atherosclerosis increases an individual's chance of cardiovascular disease and stroke. CRP is a marker for inflammation and indicate cardiovascular risk. CRP is important because it contributes some knowledge in understanding cardiovascular risk, the complement system, and detecting inflammation or infection in the body. The C-reactive protein, CRP, is annular, ring-shaped, pentametric protein found in blood plasma and produced by the liver. CRP is a member of the pentatraxin family of innate response and is a non-glycosylated protein composed by 5 23-kD subunits non-covalently associated and arranged symmetrically around a central pole. CRP is an extremely stable protein in its native form and it has a hydrophobic surface. In addition, it is a blood test marker for inflammation and rises in levels in response to inflammation. The only functions that are easily understood are the body's defenses against infective agents. When there is trauma, inflammation, and infection, then the levels of CRP increase very rapidly in response. The common causes of CRP deficiency are represented by liver failure of therapies affecting acute phase stimulus. Serum levels of CRP are increased by liver production in response to a variety of inflammatory cytokines, such as IL-6. In the reviews I read for this specific protein, the pathogenesis of atherosclerosis needs to be explained and understood before explaining the effects of C-reactive protein on cardiovascular disease. The role of CRP, other than being the first line of defense during an inflammatory response, is a correlation between atherosclerosis and cardiovascular disease that is characterized by a formation of atherosclerotic plaques. CRP values, even within the range previously considered normal, strongly predict future coronary events. CRP is the primary marker for heart inflammation and many other common diseases. The blood test, however, does not pinpoint the cause of elevation. Currently, research is being done to understand CRP more. There are papers that are continuing to research the protein, such as the role of C-reactive protein at sites of inflammation and infection, C-reactive protein and inflammation, conformational changes affect function, targeting C-reactive protein in inflammatory diseases by preventing conformational changes, C-reactive protein and the risk of cardiovascular morbidity and mortality. Research is being done by multiple scientists around the world. Macrophages secrete IL-6 and other interleukins. They bind to receptors on the liver and tell the liver that more CRP needs to be produced by binding to a hepatocyte cell. Cell-mediated immunity is triggered, so tons of CRP will be produced and increased to fight the infection, which causes an increase in concentration of CRP. If high CRP content is found in the blood, then it can be determined that the body is fighting an infection. CRP can also bind to different cell components like dead or dying cells, also known as apoptotic cells. They interact with the particular molecule of these cells, which is lysophosphatidylcholine, that are only found on apoptotic cells. They bring a complement system by recruiting complement proteins like C1q. Macrophages come to engulf these apoptotic cells to prevent the spread of infection. CRP can also adhere to bacteria cells and kill them. C-reactive protein responds to inflammation, tissue damage, and trauma, and the particular response is associated with the infectious condition. For example, CRP is highly elevated in conditions of bacterial infections. The broadest significance is to understand how CRP responds and to understand how the complement system works and whether structure and patterns are recognizable for initiation. ELISA, also known as enzyme-linked immunosorbent assay, is a plate assay designed to detect and quantify substances such as peptides, proteins, antibodies, and hormones. Proteins are immobilized on a plate and incubated overnight. After incubation and washing, biotin CRP is added to each well, and the well is incubated. After incubation and more washing, the absorbent is observed to detect protein binding. Complement activation assay. The wells are coated with specific activators. The wells are washed and incubated in a series of steps, and the detection of specific antibodies is obtained. The amount of complement activation correlates with the color intensity and measured by taking the absorbents. In this particular study, it is used to detect proteins CRP and CLP1. In other cases, phosphocholine and Alexa 55 anti-MYC antibody are added and protein binding was detected. The researchers used other proteins as the control to compare the importance of CLP1 in complement activation. The authors also used inhibitors to determine the dependency of the binding. Overall, the labels represented was what added to create the particular result and how the binding changed due to an increase or decrease of the condition of a particular substance. Overall, the labels represented what was added to create the particular result and how the binding changed due to an increase or decrease or addition of a particular substance. How CRP induces complement activation is not fully understood, and this particular study focuses on how CLP1 can interact with CRP. This particular figure focuses on inhibition and what the binding is dependent on. This is important in understanding the function of the two and the importance of the binding. It did not fully answer the specific question laid out in the introduction because it did not compare the contribution of inflammatory and infectious response. Now that they understand a little more about how and why the CLP1 and CRP bind, I would like to see how this understanding contributes to what others have researched about CRP. A possible research is to develop an understanding of how the complement activation plays a critical role in inflammatory response and the relationship to diseases. In the introduction of the paper I looked at, the researchers mentioned how CRP levels increase depending on type of infection. It mentions how CRP rapidly increases due to a bacterial infection, and CLP1 was originally found to endocytose and phagocytose gram-positive and negative bacteria. I believe this is extremely useful information because bacterial infections can commonly be treated with antibiotics. But we have CLP1 and CRP that can bind together to fight these infections. The paper gave a nice introduction about this, but the focus was more on the interaction of CLP1 and CRP versus the broader picture of how it can affect our daily lives. I would be curious to see a comparison of how the binding of CLP1 and CRP can treat a viral versus a bacterial infection. Will CLP1 bind with CRP when there is a viral infection? Or will it only bind during bacterial infection to cause complement activation? I would start with comparing the complement activation of viral and bacterial infections. Since the amount of CRP traced in the serum is different for the two, that should be researched further. Possibly even infecting a rat with a viral disease and another with a bacterial disease and taking blood samples during the incubation period, prodromal period, illness and convalescent period. In addition, using the assays used in the study to determine the binding during each period. Is the binding of CLP1 and CRP different for each period in bacterial and viral diseases? I suspect that CRP levels will be extremely high during the peak of the illness phase, and that the CRP and CLP1 binding will be at the highest because of what CLP1 was originally found to do. and how CRP seems to be already in high levels for bacterial infections.